Hello and welcome back to the Sound of Science. Today we are going to be tackling the next installment in the AP Biology series, which is Unit 2, Sections 10 and 11, Compartmentalization of Cells. Eukaryotic cells are really fascinating. They are compartmentalized in such a way that eukaryotic cells are able to have several enzymatic reactions occurring at the same time without those reactions interfering with one another. Now, all cells have a plasma membrane. This includes prokaryotes like bacteria, and it separates the extracellular from the inner section of the cell. And this allows all cells to maintain some level of homeostasis. They're able to keep that stable internal environment because of this plasma membrane. But the fact that eukaryotic cells can literally have multiple internal environments is an amazing ability, and it's what allows us to have these specialized cells and allows our cells to do these different specialized jobs. These membranes help prevent reactions from interfering with each other, and they help prevent competing reactions. So one of the examples I like to give is lysosomes. Lysosomes are used in digestion of old cell parts. They're used to break things down. So lysosomes are filled with hydrolytic enzymes, and these are the enzymes that are used in digestion and breaking down. But for the hydrolytic enzymes to function, they have to have an acidic environment. And as you know, part of maintaining homeostasis is maintaining a neutral pH, a neutral environment inside the cells. So these lysosomes actually have an acidic environment so that hydrolytic enzymes can function, but it doesn't impact the remainder of the cell, allowing the cell to maintain that neutral environment. Mitochondria have folded inner membranes, and these folded inner membranes called cristae allow for maximized surface area. Now, we've talked about the importance of surface area to volume ratio, and we've talked about how metabolic reactions occur on the surfaces of these membranes. So having many folded membranes actually increases the area for these metabolic reactions to occur. And in the case of the mitochondria, this increases the amount of electron transport that can occur and increases the amount of ATP synthesis that occur, thus allowing for more ATP to be made. If there is some type of damage to mitochondria and they don't have these highly folded membranes, we're not going to have the energy created that's necessary for life, and this actually leads to things like mitochondrial disorders. Chloroplasts are another example of these inner membranes. Instead of having folded inner membranes, they actually create multiple stacks of discs called thylakoids. And these membranes allow for light-dependent reactions and photosynthesis. So having the thylakoid membranes, which also happen to contain the chlorophyll, which is that pigment that helps capture light energy, this actually increases the efficiency of photosynthesis. So how did all of this compartmentalization begin? Because if we think back evolution-wise, life on the planet starts as single-celled organisms, most likely bacteria, of course, right? And they don't have any of these things. And in fact, if you do a comparison now between bacteria, prokaryotes, and eukaryotes, you can see the difference. You can see all of the membranes that are located in these eukaryotes, the mitochondria, and of course chloroplast, if it's a photosynthetic eukaryote. We have that nuclear envelope or nuclear membrane. We have ER, we have Golgi apparatus, all of those things. If we look over at the prokaryote, which is to the right here, you can see that it has a nucleoid region where the genetic material is contained, but that's about it. That's the only membrane, the only membranes that we see are the plasma membrane. But eukaryotes have all of these different compartments. So then how do we go from prokaryotic cells to eukaryotic cells with all of these different membrane-bound organelles? The nucleus in the ER, we theorize, formed because the plasma membrane folded in on itself. Those infoldings created these structures. And over time, those structures would have obviously been very useful. And so they would have continued. It would have occurred as the result of some sort of genetic change, right? Some sort of mutation, as most things evolve. The mitochondria, on the other hand, we think arose from an endosymbiotic relationship. There's this free living aerobic prokaryote. Remember, aerobic means it uses oxygen, and it gets engulfed by an anaerobic cell with the process of endocytosis, that active transport method that we've already talked about. When it got engulfed, when it was eaten, so to speak, or consumed, instead of being digested, it ends up with this membrane around it. So you have this bacteria surrounded by another membrane. And ultimately, it ends up supplying energy to what has now become its host cell. That's beneficial. And as a result, we end up with this structure that has both an inner and an outer membrane, like mitochondria. Chloroplasts, we believe, had a similar evolution. They occurred kind of the same way, the same sort of idea. Only the difference was with the chloroplasts, they were able to do photosynthesis. They contained chlorophyll, that pigment, in their plasma membrane to begin with. And this theory has more evidence than just like, oh, it seems like a cool theory. As you know, in science, we always back up our theories with evidence and with testing. And here are some of the things that we know for a fact that really gives evidence for this theory. So, why do we think that chloroplast and mitochondria had to arise from an endosymbiotic relationship? Both mitochondria and chloroplast have double membranes, meaning that they would have had a membrane, and then when they were swallowed or engulfed, they ended up with two membranes. And that is what we see now. Both have circular DNA, just like prokaryotes have. Both reproduce by similar processes like bacteria do. And both have their own ribosomes, that other thing that we find in common between eukaryotes and prokaryotes. So it would make sense that each of them would have both DNA and ribosomes, which is what we see. Hopefully all of this has made sense today. If you have questions about how each of these organelles functions, you're certainly welcome to go back and look at a previous video. And as always, I'll see you next time on The Sound of Science.